In this section, I'm going to talk about an introduction to bacterial growth and start by talking about inoculation. So the 5 I's represent 5 basic techniques to manipulate, grow, examine, and characterize microorganisms in the laboratory: inoculation, incubation, isolation, inspection, and identification. I'm going to provide brief definitions of each one of these now, and then we'll talk more specifically about them in detail as these lectures kind of progress. So inoculation. Inoculation is the transfer of bacteria into new media. When we inoculate bacteria into a media, it allows them to grow. Incubation is allowing bacteria to grow at particular environmental conditions. If we want bacteria to grow in a laboratory, we would put it in an incubator that's set to a certain temperature and certain oxygen requirements to support their growth. Isolation. Isolation is the separation of species in a mixed culture. So oftentimes when we get a patient specimen, that specimen is full of many different species. If you think about a nasal swab, all the different species of bacteria that could be present in your nose, or if it's a fecal sample, all the different types of microorganisms that could be present in the stool. So when we have isolation, we need to separate those apart from each other so we can start to figure out what's actually causing the infection. Inspection and identification go hand in hand. Inspection is when you observe test results of a microorganism, and identification is naming the genus and potentially species name of the microorganism based on what you saw during inspection. So this is a summary of those, and you can read those definitions that are listed here. So take a minute to pause the video and read through this 5 I's sheet. During inoculation, you want to produce a culture. A culture is a propagation of microorganisms with various media. So you use media in order to grow a culture. Medium, or plural, media, is a nutrient-containing environment in which microbes can multiply. Inoculation is the introduction of microbes into or upon media for culture. And that's what we're going to talk about during this segment, is the different types of media that we would inoculate microorganisms into. Sterile means that it's free of all life forms, including spores and viruses. It's a requirement for any instrument used for sampling and inoculation. So if we're going to try to produce a culture, and we want that culture to be relatively pure or not contaminated with other types of microorganisms, it could be a mixed culture but not contaminated with outside other organisms, then we want to use sterile equipment to transfer and grow that organism. So the media would also have to be sterile. So we're going to start talking about inoculation. So media provides nutrients in the laboratory. Microbes must be provided with all of the required nutrients in an artificial medium. Some microbes require only a few simple inorganic compounds for growth. Others require a complex list of specific inorganic and organic compounds. Certain microbes cannot be grown on artificial laboratory media and require cell cultures or host animals. So you can't just throw any bacterium on a plate and expect it to grow. Some bacteria are tougher to grow than others. Some bacteria have very specific requirements that you have to add certain nutrients to the media in order to get them to grow. This is a constant struggle because we observe microorganisms growing on a plate and we know it can happen, but providing them the correct nutrients in order to get them to grow can be tricky. So we'll talk about the different types of media and what they're used here, but know that sometimes it's difficult to provide every microorganism everything that it would ever need in order to grow. So there's things that we can try to do to get it to grow in a laboratory. Bacteria such as chlamydia can't grow in standard media. Chlamydia is an obligate intracellular pathogen, which means that it has to grow inside of cells. So in order to grow chlamydia in a laboratory, you would have to grow it in cell culture. So this last bullet point here, they require cell cultures. You would have to provide cells for the chlamydia to grow in. The good thing is we typically don't want to grow chlamydia in a diagnostic microbiology lab. That's a pathogen that we don't need lots of in order to diagnose it. We can diagnose it using simple DNA typing. The reason for growing chlamydia, though, would be in like a research setting. If you want to grow lots of it and observe things about it or try to treat it with certain things to identify new antibiotics, that would be a reason to want to grow it. So we still need to know how to grow it, but medically speaking, we don't really need to culture it in a laboratory to diagnose a patient with chlamydia. And some microbes need host animals. Borrelia burgdorferi grows well inside of mice, so we can inject it inside of mice and grow lots and lots of that organism inside of mice. Mycobacterium leprae, which causes leprosy, can't grow in media. We haven't been able to artificially grow it in a laboratory so far. Media is classified according to 3 properties: the physical state, whether it's liquid, semi-solid, solid; chemical composition, whether it's chemically defined or complex; and functional type, whether it's general purpose, enriched, selective, differential, anaerobic growth, specimen transport assay, or enumeration media. So first we'll talk about the different states. As far as chemical composition goes, I'm not going to cover that too much. You can have chemically defined. Basically, that means that every single amino acid is defined in the media list, or complex. Substances such as like brain matter or heart material or brain infusion agar, those can't be defined. It's with brain tissue, they literally take a brain from an animal, grind it up, and then put it in the media. And it's difficult to quantify each one of those nutrients from the brain tissue. So some of them are too complex to define. Other ones you can define, like 3 grams of this amino acid or 4 grams of glucose and so forth. And then the functional type, we will go into that in detail. So liquid media is water-based. It does not solidify at temperatures above freezing, and they flow freely in a tilted container. Broths, milks, and infusions include liquid media. Semi-solid media are clot-like consistency. They're kind of like jello, and they're used a lot for determining motility, whether the bacteria can swim or not. Why do we care? Well, if you want to identify the bacteria, you need to make a list of all of its characteristics. Those characteristics can help us identify, especially with those enteric pathogens. If you know that it's a non-motile gram-negative rod that came out of the digestive tract, you can start to think of the species of bacteria that are not motile. It could be Klebsiella. So it can help us start to examine these characteristics in order to determine an identity. And that's really the goal, and that's what I'm going to focus on in this course, is identifying human pathogens and trying to figure out what is the causative or infectious agent. Solid media provides a firm surface upon which cells can form discrete colonies. It's used to isolate bacteria and fungi. Agar is commonly used in solid media. It's a complex polysaccharide that's isolated from algae. It's solid at room temperature and liquefies at 100 degrees Celsius. So you can put it in a incubator up to that, and it's not going to liquefy. So it stays solid at a wide range of temperatures. Any medium containing 1 to 5% agar usually has the word agar in its name. So this is a picture of the various types of media. So we have liquid media, semi-solid, and then solid. So solid is not going to change shape if you move it on its side. There are 2 main growth patterns that you would see on solid media: a lawn and a colony. A lawn probably reminds you of grass, and that's exactly what it looks like. So a lawn where you have grass is everywhere. So the plant grass is all over the place. It's continuous growth in a section. That's what a lawn is. So lawn is bacterial growth that covers an entire surface. There's no kind of breaks in that growth. It's just a solid surface of growth. A colony is a discrete mound of cells. So this little dot here is a colony. A colony contains cells that arose from the cell divisions of one cell. So basically what happens is when we produce a colony, one cell lands on the plate, and then it divides and divides and divides and divides and divides until it gets big enough for us to be able to see with the naked eye. Every single cell within a colony is definitely of the same species, and it's extremely genetically close in genetic composition to all of the other cells in that colony because they arose from the cell divisions of one cell. Now moving on to liquid media, so there's different growth patterns that you can see in liquid media. Uniform turbidity means that it's cloudy throughout the tube. So you have equal cloudiness throughout the tube. Flocculent is referring to clumps of growth. So you have clumps growing throughout the tube. A pellicle is a film of growth on the surface of the broth. And sediment is when bacteria settles to the bottom of the tube. So a majority of the growth that you would see would be in a cluster at the bottom of the tube. Again, why do we care about this? Well, if you want to identify a microorganism, you need to be able to make note of all of these characteristics. If I were to see a tube that was isolated from a patient's respiratory tract, and in that tube, I have a specimen, I know it's a pure culture, and there's a microorganism that's forming a pellicle, and the pellicle looks really slimy. It's almost brownish green in color. And then there's some growth throughout the bottom of it. There's some turbidity, but that pellicle is there and it looks really slimy. I'm going to start to think that it's a Pseudomonas infection because that's what that looks like. If you grow it in a broth tube, you can kind of get a feel for how these microorganisms look. And microorganisms are going to grow pretty similar within the same species. So if I know Pseudomonas is one of those that's going to form a slimy pellicle, and I see my patient's sample forming a slimy pellicle, I can start to think about what this organism might be and provide a presumptive diagnosis. Remember the difference between presumptive and confirmatory diagnosis. Presumptive is an educated guess. We think we know what it is. We think it's this general genus of bacteria, while a confirmatory diagnosis has been confirmed by at least 3 different methods, and we know pretty much for sure the genus and species that's causing the infection. Now this is a picture of various growths, and this actually came from my first master's degree thesis, where I was identifying certain characteristics of Lactobacillus calleohominis. Now Lactobacillus in general is a genus of bacteria that grows in the digestive tract and in the vagina and various other locations in the body. It's a good guy. It's a beneficial bacterium that produces acid in order to deter the growth of other microorganisms. There's all different species. There's Lactobacillus acidophilus, which is most common as a probiotic. You can find it in many different types of yogurts, and it's a good guy in the gut. And then there's some that are kind of specific to the vagina, like Jensenii, crispatus, Gasseri, Einers, and then this new one, calleohominis. So I was growing them in different tubes to try to see general characteristics of these guys. And you can see acidophilus definitely forms a sediment. Jensenii has uniform turbidity. And if you look at the 2 different strains of Jensenii, you can see that both of them have uniform turbidity. Lactobacillus crispatus is one of the highly protective microorganisms, and the CTV05 is a capsule of probiotic that can be, or used to be able to be prescribed to recolonize the vagina with good bacteria. It's really strong, tough, and forms a really impenetrable barrier to prevent chronic infections in the vagina, such as like Candida albicans yeast infections or bacterial vaginosis that's caused by Gardnerella vaginalis and other types of infections. It can even protect against STDs, and it has been shown to deter the penetration and infiltration of HIV in through the vagina, so it lowers the risk of HIV infection. So this one's a pretty tough microorganism, and you can see how it's not just forming a loose sediment, it's actually forming an aggregate, and you can see how it's kind of clustered together, and it's so tightly aggregated together, and you can see that not only in the clinical strain or the strain that we would give as a probiotic, but also in the clinical strain. You can see that same type of pattern appearing here. Gastri kind of forms a biofilm type growth along the side of the tube, so you can see that happening in both of the strains of Gastri. And then you can see Einer's is all going to be that uniform turbidity, just like Jensenii. And this new species, Lactobacillus calleohominis, actually started to form flocculent growth. And those were characteristics that you make note of. Again, not only for identification, because that's really important, like I keep mentioning, but also knowing how they grow in a tube may give us clues how they grow in the body. You can see how these are all weakly associated. So Einer's usually takes over when someone's pregnant. That becomes the predominant probiotic in the vagina there. It's a very weak colonizer, and it's weakly protective. So pregnant women usually tend to get more infections than someone that's not pregnant. And that's all due to hormonal changes and so forth. And plus your body is preparing for a baby to go through the birth canal. So you don't want to have this super tough and harsh acid producing microorganism present when the baby is going to be going through the birth canal. When you look at calleohominis and you look at this clustered growth, you can start to make some assumptions about how this would colonize the vagina. Now, it's not going to be as tough as this one, but you can definitely see that with these clusters, it has the ability to form aggregates. So it might be a pretty good protector in the vagina. Since my research here, not much new information has been published about Lactobacillus calleohominis or its protective features. We have Crispatus CTV05, and that's kind of the standard that they use to recolonize women who have chronic infections. General purpose and enriched media. So general purpose media grows a broad spectrum of microbes. Enriched media is general purpose plus nutrients. So added nutrients are in this media in order to grow fastidious bacteria. So fastidious means that they're difficult to grow. Nutrient agar, this is general purpose media. It contains nutrients to grow a broad range of microorganisms. Chocolate agar is enriched media. This grows fastidious microorganisms, and again, fastidious microbes do not grow on standard media. They require extra nutrients in order to grow. Chocolate agar consists of boiled blood. So it's blood cells that have been boiled and broken open, and that's why it looks like brown in color. Differential media allows organisms to grow in a different color. So depending on the species characteristics, different species will grow different colors. This is a picture of a chromogenic agar, and you can see all of the different colors that the different species are going to grow. Different nutrients can be added with certain indicators which allow for these various color changes. There's something called agar art, and if you have a free moment, you should go look up agar art, especially by the Society for Microbiology. So this agar art, they have like competitions with it. They'll actually draw really cool pictures on the surface of agar plates, on chromogenic agar, using different species of bacteria. Excuse me, those bacteria will grow in different colors, so it makes for really interesting pictures. So go ahead and look up agar art if you have a moment, and you'll be amazed at how nerdy microbiologists can be. Differential media, blood agar is a good example of a differential media. So there's hemolysins, that enzymes that lyse or break down red blood cells to release iron-rich hemoglobin. And there's different versions of hemolysis based on what that microorganism produces. So beta hemolysis is complete lysis of the red blood cells. Certain species are beta hemolytic. That means that they produce enzymes that can completely digest red blood cells. Alpha hemolysis is incomplete lysis of the red blood cells. So certain species produce enzymes that partially lyse red blood cells. And gamma hemolysis is no hemolysis. That means that the microorganisms do not produce the enzymes in order to break apart red blood cells. An example of a beta hemolytic bacterium would be Staphylococcus aureus. An example of an alpha hemolytic bacterium would be Streptococcus pneumoniae. And an example of a gamma hemolytic bacterium would be Staphylococcus epidermidis. So different species have this ability to either digest or not digest red blood cells in the media. This is a picture of some plates with examples of hemolytic and non-hemolytic bacteria. Enterococcus does not digest the red blood cells, so that is gamma hemolytic. This one does partially, so you can see alpha hemolysis occurring here. And then these two you can see straight through the agar plate, so those are beta hemolytic. Selective media selects which organisms can grow and which organisms cannot grow on its surface. So selective media selects against the growth of certain microorganisms. An example of a selective media would be colistin nalidixic acid, CNA agar. CNA agar kills off gram-negatives, so it selects against gram-negative bacteria, while encouraging the growth of gram-positive bacteria. This is a good agar to use in skin infections. So if we want to selectively grow gram-positive organisms from a wound culture or something on the surface of the body, we can grow it on CNA. CNA is going to kill off the ones we don't care about, leaving the gram-positive skin ones that are probably causing the infection. There's some media that are selective and differential. They're designed for special microorganisms, and they have extensive applications for isolation and identification. McConkey is an example of a selective and differential media. It's selective because the bile salts and crystal violet select against gram-positives. So only gram-negatives typically grow on this one. It's also differential based on a neutral red indicator. It's a pH indicator. If the bacteria have the capability of producing lactose, then the bacteria will grow pink on here. If the bacteria do not have the capability of fermenting lactose, then you're going to see that they turn colorless. So here you can see the lactose fermenters on this side and the non-lactose fermenters on this side. McConkey agar is predominantly used for enteric pathogens, those pathogens that grow in the digestive tract. In the digestive tract, you have a lot of probiotics, a lot of good guys, and those good guys are usually gram-positive organisms. So we grow it on McConkey because McConkey kills off the gram-positive organisms, those good guys. We don't care about them in this instance. We care about the bad guys, the ones that are causing the infection. So kill off the good guys, that eliminates some of the growth on the plate, and then you leave the bad guys for us to investigate. It's also differential based on lactose fermentation. So if it is a lactose fermenter, it could potentially be E. coli. If it's not a lactose fermenter, it could potentially be Pseudomonas. Reducing media is used to grow anaerobic bacteria. Carbohydrate fermentation media allows us to determine which sugars the microorganism can utilize, and this is all for reporting characteristics for diagnostic purposes. Transport media allows us to transport microorganisms. Assay media is used to test the effectiveness of certain drugs. It's used to assess the effect of disinfectants, antiseptics, cosmetics, preservatives on the growth of microorganisms as well. And then there's enumeration media, which is used in industrial and environmental microbiology to count the numbers of organisms in milk, water, food, soil, and so forth. So enumeration media allows us to do cell counts.